And that's what happens with cholesterol in our treatment. For so long as we keep the liver at a cholesterol deficit, the rest of the body starts returning cholesterol to the liver. You get a suppression of inflammation, you get a suppression of all the horrible things that are happening because you had too much free cholesterol throughout your body. And the consequence of real interest is that atherosclerotic plaque starts very rapidly regressing and shrinking and becoming more stable rather than fatty and unstable. And that's great.
Hi, everybody. My name is Jose Navarro Betancourt, and I am the scientific director of quadraScope. We invest in a broad range of technologies that enable disease treatment and prevention through biological age reversal. And today, I will have a conversation with Reason, who is the co-founder and CEO of one of our portfolio companies, Repair Biotechnologies. Reason, thank you very much for taking the time to talk with me.
Happy to do so.
So, Reason, let's begin with a quick overview of Repair Bio. What is Repair Bio's mission?
So when we founded it, it was to do something that's in the name, find something that's damaged that happens with aging and repair it, and therefore turn back the progression of degenerative aging somewhat. Right now, our focus is on atherosclerosis. We have a technology that enables us to reverse that condition, which is currently very hard to do. So we're leading that off to clinical trials. We've been developing this for the last 4 years. We're quite close to getting our IND in within the next 18 months at this point.
Why is cholesterol degradation so important for aging? What is atherosclerosis so important? Obviously, it's a very widespread disease, but what are the challenges with cholesterol degradation?
Right. So our technology obviously breaks down cholesterol in a very specific way. I think everybody understands by this point in time, after half a century of being told about it by doctors, that the LDL carried cholesterol in your bloodstream is important over the long term, and therapies try to reduce that. But unfortunately, that doesn't have a great deal of impact on local aggregates of cholesterol that occur throughout the body. Cholesterol is one of those molecules that's essential. It's needed everywhere. Every cell needs it for cell membranes and other functions. But it's only made in the liver or taken in through the diet, mostly made in the liver, and then it transports everywhere around the body. And that transport system is kind of a Rube Goldberg assembly of particles and mechanisms. And like everything complicated, it breaks down with age. And one of the consequences is that you end up with local excesses. Or if you get fat, you'll end up with local excesses of cholesterol. And cells only have a certain amount of ability to deal with that. They can put the extra cholesterol in their membranes. They can esterify it into droplets, or they can try to hand it off to particles to carry it back to the liver, but they can only do so much of this. And it doesn't take a lot of excess locally to make cells stressed and start to kill them. And that affects tissue function in many detrimental ways. So that's why you can't just target the delivery system, the LDL cholesterol coming out from the liver, and you can't just target the HDL coming back to the liver. You have to actually go after the excess cholesterol that's in a location, and that is what we do, and that makes that picking which cholesterol you go after makes all the difference. And unfortunately, it also means that cholesterol is a tough target for small molecules because you can't go after it that way.
Let's dive a little bit into the key innovation of Repair Bio, this cholesterol degradation platform that uses a new protein essentially to allow cells to degrade excess free cholesterol. And I want to really try to understand what's the difference between free cholesterol and the cholesterol, as you said, that makes part of the cell membranes that is necessary for cell functions. How do you distinguish the cholesterol that has to be degraded from the normal cholesterol?
So it's actually a little bizarre if you think about it, that cholesterol itself, the molecule, is toxic on its own. It's something that we need desperately. It's used everywhere. We have enormous amounts of it in the body, but a cholesterol molecule just floating around on its own is actually toxic. the vast amounts of cholesterol in the body are all either esterified into droplets, making them non-toxic, or they're in cell membranes where they're sort of locked into a structure that makes them non-toxic, or they're attached to transport particles to move around the body, which makes them non-toxic. But if cholesterol is sitting there on its own, unattached from any of those things, it's actually a toxic molecule. And that's kind of the interesting paradox at the heart of the whole problem, and why, as soon as you get this dysregulation that leads to cholesterol, you have problems. But we're going to talk about the origin of this cholesterol degrading platform, this fusion protein that lets you break it down safely. This actually comes out of the University of South Alabama. And like many sort of fairly radical advances, it kind of comes out of left field. It wasn't the cardiovascular people, the cardiovascular research people that were looking at this, because all the cardiovascular research people are stovepiped off into thinking about transport of cholesterol. Rather, this came out of originally the research that came into being because of the Gulf oil spills. And the researcher Richard Honkanen was thinking maybe we could make something that could do something about this. We could go look at bacteria or bacterial components or something in cells that lets you break down the components of oil. And that led to research on tuberculosis. Interestingly, because the intersection there is that tuberculosis bacteria, they can live in cells for a very long time dormant, which is why it's a difficult disease to treat relative to some other infections. And it turns out that tuberculosis bacteria are feeding on cholesterol. They use cholesterol as an energy substrate, and that's how they survive in cells, just sitting there being fairly dormant, feeding on your cholesterol. So this all in turn led to the creation of the fusion protein that's actually really human genes, because all of this looking at bacteria turned up a bunch of very interesting mechanisms, and then analogous mechanisms do exist in humans. They just don't operate in near all cells. So all cells technically have the capacity to break down cholesterol, but they don't use it. They don't express the necessary proteins. They don't, in conjunction. So the fusion protein then takes these necessary human proteins that are not expressed together anywhere in the body, near anywhere in the body, and joins them together in an optimized form that lets you then, if you put it into a cell, it does the function that the bacteria originally did, but using human genes in order to break down the excess cholesterol. And sadly, the fixing the oil spill sort of that never materialized as an outcome of all this research, but it was why it started in the first place. So like many sort of interesting discoveries in the sciences, where it started is not where it ended up. And the important advance in atherosclerosis came from nothing to do with the cardiovascular research community. And this is a story you will see repeated again and again and again, and why fundamental research is important, even if you don't know where it's going.
That's right. Fundamental research is all about out-of-the-box thinking and trying to connect the dots. Correct me if I'm wrong. Repair Bio came out of a research project from the Sense Research Foundation, right?
Research Foundation is actually one of our investors. They did introduce us to the research group that made this discovery originally. And as soon as I saw this, I said, we must do this. We must license this and develop it. This is amazing. And this was prior to any animal studies. They only had cell data at that point, but it was really amazing. It was really amazing data and an amazing piece of technology.
If we were to classify the problem that Repair Bio is solving according to the sense types of damage, we could say that Repair Bio is targeting intracellular aggregates or the buildup of intracellular unwanted products. Correct?
Yeah, absolutely. That's the general heading it falls under. And I've given presentations this year and will give presentations in December about, hey, maybe we should add excess intracellular cholesterol to the hallmarks of aging, because that it definitely shows up with aging. It's really not a good thing. And at least with the hallmarks, if you're talking about hallmarks, you don't need to say, you don't need to prove that it's a cause of aging. And clearly, I think it's not a cause of aging. It's like blood pressure. Blood pressure is very influential. It's very harmful when you get hypertension, but it's not a root cause of aging. It's somewhere downstream, but it's an important downstream effect. So you get this because other processes have been dysregulated by the damage of aging. But then the fact that you have the excess cholesterol is in and of itself harmful and contributes to further downstream problems.
So you have this humanized enzyme that can degrade cholesterol, and this is the core of your cholesterol degradation platform. So with your therapy, which cells in the body are gaining the capacity to degrade cholesterol?
Well, eventually, you want a therapy you can target anywhere, selectively, but like all gene therapies, the delivery is the whole of the therapy. Your technology always just works. You put the fusion protein into a cell, it does the job, it's not toxic, it's very safe. The trick is, how do you get it to the cells that you want to get it to? So our current therapy under development for atherosclerosis actually targets the liver very selectively because it's, quote, easy, unquote, to deliver gene therapies to the liver, or at least it's practicable. And there are very well-established ways of doing this very selectively. So if you want a gene therapy to go somewhere else, well, now you're back into development land and you have to build something. And there aren't that many other organs that you can reliably deliver gene therapies to if you're injecting it intravenously. It turns out that if you go put this drug into the liver and you degrade cholesterol, just the excess intracellular free cholesterol in the liver, and by free cholesterol, I mean cholesterol. It's just the normal cholesterol molecule, just not esterified or bound in membrane or attached to particles, then you produce tremendous benefits throughout the body. And one of the beyond the innovation of the fusion protein itself, the other major innovation that we've developed at Repair is that the liver itself, the dysfunction of the liver in obesity or aging, is contributing enormously to atherosclerosis. And this is a new discovery. This was not suspected by anybody outside of the liver disease community. The liver disease community has long thought that excess cholesterol inside liver cells was a problem. They just couldn't prove it because there was no therapy to just remove it in isolation of other changes. But the cardiovascular community has never really looked to the liver in any way other than a source of cholesterol that's getting put into the bloodstream. But it turns out that the liver's cholesterol content is affecting the body systemically in ways that disable and damage the normal regenerative processes operating in the arteries that will regress your atherosclerotic plaque. So if you can just get rid of this excess cholesterol, suddenly it kicks into place all of the normal mechanisms whereby plaque is regressed. The cells that are actually trying to repair the plaque suddenly are much more effective. And you see dramatic reductions in plaque. A very sort of simplified way of thinking about this is if you create a cholesterol deficit in the liver, then many signaling processes throughout the body dial up the transport of cholesterol from everywhere in the body back to the liver, which we then, of course, break down as it arrives. So you get this, it's like pulling the plug at one end of the bath. Pull the plug at one end of the bath, the water everywhere will head in that direction. And if you can just keep that sustained, then the bath will run out. And that's what happens with cholesterol in our treatment. For so long as we keep the liver at a cholesterol deficit, the rest of the body starts returning cholesterol to the liver. You get a suppression of inflammation. You get a suppression of all the horrible things that are happening because you had too much free cholesterol throughout your body. And the consequence of real interest is that atherosclerotic plaque starts very rapidly regressing and shrinking and becoming more stable rather than fatty and unstable. And that's great.
Yeah, let's, plaque regression is basically the goal of atherosclerosis as a therapy, right?
It's the holy grail.
the Holy Grail, right? But let me just wrap up this idea of the liver kind of as a cholesterol sink, right? So you give the liver the ability to very rapidly degrade cholesterol, and then the cholesterol starts flowing into the liver. And I guess there's also an endocrine function of the liver that actually secretes some protective proteins, right? That tell the endothelium in arteries that it should stop accumulating cholesterol, I guess, or it tones down inflammation in the vascular endothelium. Is that a good way to say it?
It will be the work of decades for researchers who come after, the first approval of our therapy to try to work out how is the liver communicating with the body in order to achieve these benefits. Fortunately, we don't have to prove any of that.
Mm.
We just have to show that yes, it's upregulating the transport of cholesterol back to the liver. And it's also reducing inflammation. We don't have to say how it's reducing inflammation, which is good because we don't know. We don't have the full understanding of why that's happening. But all of these effects join together to produce an improvement in health throughout the body. And the more you get rid of the excess cholesterol everywhere in your system, the better off you are.
And if you can even reverse atherosclerosis, plaque progression, that's even better. As you said, the Holy Grail. So
Yes, because it can't be done at present.
that's right. But let's dive into this cholesterol regression, atherosclerosis plaque regression a bit. You obviously know that atherosclerosis plaques have a progression through different stages. They begin with lipids only intracellularly in cells. Then lipids start getting accumulated extracellularly, and then you start getting fibrosis on the plaque, and then even calcification. So, what is the best stage for repairs intervention? I'm assuming that it's when lipids are only intracellularly, correct?
Well, in principle, you could use the therapy at any point and it would improve things. In practice, using mouse models, that's next to impossible to demonstrate because you don't really have a way to, if you're looking at mice which only have fatty streaks in their arteries, the very earliest stages, trying to assess whether you're reversing them, you're kind of bouncing off the baseline of measurement. It would be very hard to do. So all of our studies in mice use very severely atherosclerotic mice, where they have big plaques, and it's very easy to see the reduction in size using the histological approaches we take to that. Now, once you get into people or even non-human primates, but the non-human primate studies will be also looking at monkeys that are as atherosclerotic as one can make them, so you can see a large effect. In principle, it should be possible to put this therapy into a person with fatty streaks and see those fatty streaks reduce. And in humans, we have the imaging technology to actually go in there and really do that and see that. But that won't be the first focus because it never is. All the incentives for development of medicine mean you have to go after the severe forms of the condition first, because that's where people are willing to pay money. And that's where the regulators are willing to approve therapies. Regulators are very unwilling to approve. But strangely, atherosclerosis is one of the only areas of modern medicine where prevention is the primary focus. It's ironic, but generally modern medicine is really not about prevention, and it's very hard to get therapies approved for prevention because the regulators are very loath to allow you to treat, quote, healthy, unquote, people. So all of the incentives line up to mean you treat the severe cases. So we go after the mutants who have accelerated atherosclerosis and large lesions as soon as you find them. We go after people who've just had a heart attack because they realize they have heterozygous familial hypercholesterolemia, and we go after the severe cases where people will die in the next year or two, and there's really very little that can be done for someone who's in that state with the present standard of care. So you start with the severe cases, and then over the next 20 to 30 years, you would expect, as the therapy evolves and as the medical community starts working with it and figuring out what the bounds of the possible are, you will see people using this in the earlier stages, and you will see studies in the earlier stages. And my vision for this is, let's say 50 years after first approval, whatever the ultimate form of this therapy happens to be, people will use it starting in their 40s, once every 5 years or so, and they will never develop atherosclerosis. But obviously, that's a long way down the line, and first things first.
Yeah, I mean, prevention is key for atherosclerosis because since it's a root cause of disease, atherosclerosis is the cornerstone of stroke, heart attacks, and other deadly conditions. Let's dive into familial hypercholesterolemia, which is an inherited condition caused by mutations that impair the body's capacity to eliminate cholesterol from the blood. This is a serious condition that leads to accelerated atherosclerosis and death from cardiovascular diseases. Familial hypercholesterolemia is obviously very challenging to treat. How are these patients treated today? What are the options for these patients right now?
So the homozygous familial hypercholesterolemia condition, we'll call it HoFH because it's just a pain to say.
That's right.
And HeFH for the heterozygous version. So the homozygous version, you have mutations in both copies of the LDLR gene, which is the gene that the liver uses to take up cholesterol. So your bloodstream has an enormous amount of cholesterol in it, and over time that really increases your risk of atherosclerosis. These patients, if they were untreated, they die in their 30s. From heart attack and stroke, because they have so much atherosclerotic plaque developing early in life. Now, the current treatment options are not great. There are permanent PCSK9 or semi-permanent PCSK9 inhibitors you can take, where you'll take someone with a blood cholesterol of 1,000, and they take it, and now their blood cholesterol is 500, and a normal human is, under 100, you'd hope. So 500 is better than 1,000, but it's still, it's not fixing the problem. And these drugs are enormously expensive to the payers, obviously. Now, the thing that they more commonly do is apheresis. These people go in and get apheresis of their blood to remove LDL cholesterol, once every couple of weeks, and that will run you $250,000 a year. And that doesn't really, enormously, it's managing, it makes things better, but it's not solving the problem. They still die young, and they still die from heart attack and stroke. So you need a way to regress the plaque, which is what we don't currently have, because lowering LDL cholesterol does not regress plaque. It doesn't solve the problem of the plaque being there. It doesn't make it go away. It just slows down the development of that plaque. And obviously, if your option is, well, I can reduce my cholesterol down to merely 3 times human normal, then slowing becomes a bit of a joke at that point. You die in your 50s, not your 30s.
Yeah. Let's dive into this concept of regressing the plaque a little bit more. There are some reports that high-dose statin therapy can regress the plaque from 5 to 15%. Obviously, this is not a lot, but how do you explain these results? What's happening there?
So, I mean, let's accept that there's an enormous variation in the human population in terms of, the genetic variants that contribute to your cholesterol metabolism and how you will respond to various different interventions. If you look at the really big meta-analyses of statin use in the population, I believe that you have 4 years of statin use or 18 months of statin use, something along these lines, a long time of using it, and then look at what plaque is doing. The range is something like -3% to +5% plaque volume. So clearly some people do get plaque reduced by a little bit, but the average is about 0. But because the average is 0, obviously that means some people are getting small reductions in plaque, but they are small. The interesting thing is that even very small reductions in plaque volume produce very large reductions much larger reductions in risk of stroke and heart attack. I think the numbers from a big meta-analysis were that if you're one of the lucky people who uses statins for like a few years and gets a 1% reduction in your plaque volume, just 1%, you get something like a 20% reduction in risk of stroke. And the Ornish diet does better than statins, but even there, it's a very hard diet to adhere to. You'll get a reduction in stroke and heart attack incidence, but you're not getting your plaque regressed. So Ornish diet patients in the study, they pretty much all had cardiovascular events over the course of the study, just less than the people who were not on the Ornish diet. And it's worth noting that, like, okay, statins, the numbers are 10 to 20% reduction in risk. Now, if you're an epidemiologist, 10 to 20% is great. That's a big number. If you're a person looking at your risk, 10 to 20% is nothing. That's not a great number. That is, you are still going to get a heart attack. So yes, every boutique cardiology practice can roll out an example of a patient who got 10% regression on their their combination of PCSK9 inhibitors, diet, statins, etc. But ask them what the average is, because the average is 0.
Statins were for a while one of the top selling drugs worldwide, right? I believe Lipitor was a top selling drug for many years in a row. So this really emphasizes how big the atherosclerosis problem is, and what's the potential for
I mean, the cost of treating atherosclerosis, not the full opportunity cost of suffering the condition, but the cost of cardiovascular disease over a lifetime is $750,000. That's the cost to treat, to payers, not necessarily the individual. And that is why these drugs that have, only very modest effects on risk are such big sellers, because the payers desperately want to reduce that $750,000 cost per patient as much as they can. But again, as an individual, 10 to 20%, that's not great.
I think this is a good point in the conversation to bring up again the PCSK9 inhibitors that you touched on very briefly. So to set some context, PCSK9 is an enzyme involved in the degradation of LDL receptors in the liver. So lower levels of PCSK9 allow the liver to clear more LDL cholesterol. There are now some medications that are monoclonal antibodies to inhibit PCSK9. You have Praluent by Regeneron, you have Repatha by Amgen that are FDA approved. You also have Leqvio by Novartis, which is siRNA. And you have Verve Therapeutics, which is even base editing gene therapy to inactivate the PCSK9 gene. So how does Repair Bio compare with this next generation of therapies that in theory can reduce cholesterol really effectively?
Yeah, I mean, the studies have shown that you can reduce cholesterol down to 10% of human normal. And I think they're starting to think that this will have negative long-term effects on the brain, but it's still too early to really say that. But there's a reason that there's a reason we didn't evolve to have cholesterol of 20 rather than, let's say, 80 or 100. We just don't know what that reason is, because in the short term, humans seem perfectly capable of being very healthy with a cholesterol of 20 rather than 60 or 80. So PCSK9 inhibitors in principle allow you to slow the development of atherosclerosis as much as it is possible to do with lowering LDL cholesterol. The problem is that you can't get rid of it entirely. You can get to the point in the present situation where you will die from cancer before you die from atherosclerosis. But if somebody solves cancer, guess what? You're still going to die from atherosclerosis somewhere down the line if lung disease doesn't get you or something else. So lowering LDL cholesterol doesn't solve the problem. it really doesn't solve the problem. Because it doesn't get rid of the plaque. There's nothing you could do to LDL cholesterol that will regress existing plaque because it just sits there. Because okay, so you've taken away some of the input, but that doesn't prevent the plaque from continuing to be a toxic lump of free cholesterol that resists the body's efforts to get rid of it. In part because the liver has also got too much cholesterol in it. And PCSK9 inhibitors aren't going to help that situation because they're encouraging the liver to take up much, much more cholesterol. So it's a tough problem. PCSK9 inhibitors are definitely, an improvement over statins, and ultimately they may supplant statins to the extent that they become cheaper. But they don't do anything. Any incremental development that is a better statin, it'll make the developer a lot of money for the reasons we've discussed previously. And certainly everybody in the industry looks at statins and doesn't see a tremendous failure. They see dollar, 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 dollar. So it continues to be the case that new statin-like things are very popular in the development circle, but it doesn't solve the problem. It just makes money.
Let's dive a little bit into Cyclarity. Cyclarity is a company focusing on degrading oxidized cholesterol specifically. Is there a role for this degradation of oxidized cholesterol as a complementary therapy to Repair's approach?
So Cyclarity is a really excellent example that sometimes you don't need animal studies to convince regulators to let you put something in humans. You just need something that has a very long history of being pretty safe. And that's just as well because in Cyclarity's case, they're targeting 7-keto cholesterol specifically, which is one specific rather harmful altered form of cholesterol that has no useful purpose in the body. And so getting rid of it is great. And they produced a cyclodextrin, and cyclodextrins are well known, well established, very safe. They produced an altered form of cyclodextrin that combines specifically 7-keto cholesterol, so you can just throw it into the body and clear out 7-keto cholesterol, and hopefully that produces a good effect. But there are no animal models that really reflect how it would work in humans because the animal models will not have the right amount of 7-keto cholesterol in the right places. So you can put it in animal models and show that it does the thing you expect it to, that it goes and finds 7-keto cholesterol and pulls it out, and they've done that, but you can't draw any conclusions about its efficacy. So the standard challenge in aging is that all mechanisms exist, but we have no idea of their relative importance. So 7-keto cholesterol almost certainly is a contribution to all sorts of things, not just the few rare conditions where it's absolutely certain that it's the problem. So it almost certainly contributes to atherosclerosis, but is it a large part of the problem or a small part of the problem? The only way you're going to find out is by putting it into non-human primates or humans. And since they're using cyclodextrins, they can just go pretty much straight to humans, which is what they're doing. So the first thing we're going to find out regarding whether it works is in their phase 2, which is an interesting sort of backwards way of going about it for most biotechs, because in most cases you have to show things work in animals before you get to somewhere, because you're using a new drug that hasn't been done before. Therefore, you have to show all sorts of safety and efficacy to convince people to come along. But this is one of those rare cases where if you have something that's already known to be very safe, you can do this sort of let's just do something much more like the cosmetics pathway, where you're just running straight into humans to demonstrate the point, which is a good thing in this case, because it's probably going to take 10 years to manufacture animal models that look correct for 7-keto cholesterol levels, and nobody's going to bother to do that unless 7-keto cholesterol is in fact relevant in the human condition. So we actually at Repair, our therapy does break down all altered forms of cholesterol. It breaks down 7-keto cholesterol, oxidized cholesterol, acetylated, et cetera, all the modifications that one sees on cholesterol, they're also broken down by our approach. The question, it is a question as to whether 7-keto cholesterol is very important versus just a contribution, because you can put you can put normal cholesterol, just straight free cholesterol on cells in culture, and the cells will become very unhappy. It just takes more of that cholesterol than the case for 7-keto cholesterol. It takes much less 7-keto cholesterol to make the cells very unhappy. And the same for the oxidized cholesterol, it takes less of that. Acetylated cholesterol takes less of that. But the plaque has no shortage of cholesterol. My concern with their approach is that, look, a plaque is a sea of cholesterol. There is more than enough cholesterol in that plaque to produce all of the problems that are keeping it being a plaque. And same for the liver, a fatty liver. There's more than enough cholesterol in there. 7-keto cholesterol may be making things worse. We don't know how much yet. So we'll see. And I wish them the best of luck.
I guess only time will tell.
Yeah, exactly.
Right.
And kudos to them for managing to raise the money and getting to the point they're at with entering human trials this year.
That's right. And I wanted to talk briefly about GLP-1 agonists, since everyone's talking about this, and there's actually some reports showing that suggesting that GLP-1 agonists might slow down atherosclerosis by reducing endothelial inflammation. So is it possible that the widespread use of these drugs for obesity will make a significant impact in the prevalence of atherosclerosis in the next decade?
Prevalence, no. Severity, yes.
Hmm.
So atherosclerosis is near universal. It's the very rare person who gets to 60 without, meaningful plaques. If you're obese, everything is worse. Obesity makes everything worse. But for all the obvious reasons, for the mechanisms that we care about, where there's just excess cholesterol everywhere, and for all the other things like inflammation and dysregulated, everything to do with diabetes. Diabetes messes up the whole of the body. So obesity just makes everything worse. So to the extent that you can get people to lose weight, hey, everything got better. So you're starting to see very disingenuous studies out there in the world of all the current weight loss, the weight loss therapies, where they're saying, hey, this is an amazing thing. Look at what it's doing. It's like, hey guys, they're losing weight. It's obviously going to be great compared to having excess weight, because excess weight is terrible. And that's that story. That GPL1, the GLP-1 agonists are not, the second coming. They're just a way to lose weight, and weight is terrible. So don't be overweight.
To summarize, you told me you are 18 months away roughly from an IND. You are targeting homozygous familial hypercholesterolemia as a lead indication, and you're going with gene therapy for the liver using mRNA gene therapy, correct?
Correct. Lipid nanoparticle delivery of mRNA, which is just a very short-term treatment. So you go in and you dose every week, and you get a few days of the effects of clearing out cholesterol. And doing that is enough to produce these profound, for like 6 to 8 weeks, is enough to produce profound effects in the animal models. So that leaves you with a therapy that has nothing left afterwards. It's just like an expensive small molecule. You take it, it has an effect for a little while. If you want more, take it again.
Is there a role for perhaps longer acting gene therapies, for example, AAVs, or cell therapy?
Yeah, maybe. AAVs are very out of favor right now for high dose therapies. And certainly you're not going to find support in it from investors or the FDA. Maybe more investors are the other roadblock here than the FDA itself. You're not going to find support for using high dose AAV for any that goes to the liver, because there was a study a few years back in which people died. And that basically has shut down near all efforts to use AAV at high doses that go anywhere near the liver. So that's kind of out of the consideration right now. Cell therapy, yes. The chief objection to cell therapy is that it's very hard. It's very hard to do that. Even very large entities have struggled with forms of cell therapy. But if we were going to do cell therapy, we would make macrophages and we would give them our CDP technology and then introduce them into the body in very large numbers. And off they would go and do their job. But it's very challenging. It's a lot of expense and trouble and pain to get to that point. Generally, cell therapy companies need very high capitalization, and even those with billions have failed in trying to do what they were trying to do. So yes, in principle, no in practice.
And atherosclerosis is such a prevalent disease that you need something that can be mass produced relatively easily as mRNA.
Yes, and mass production of cell therapies is in the future. We're
Right.
using universal cell lines, but the road to universal cell lines has all of the cell therapy disadvantages attached to it, in that everything is difficult, everything is expensive. It's very challenging.
And talking a little bit more about the future, what is next after treating atherosclerosis? And I specifically want to talk about the role of cholesterol metabolism in Alzheimer's disease. Is there a role for this cholesterol degradation platform in Alzheimer's?
No, I think so. I mean, all of the neurodegenerative conditions, you're clearly seeing dysfunction of lipid metabolism. Again, we come back to all mechanisms exist, but it's unclear as to which of them are the most important, and that's especially true in Alzheimer's disease, a very complicated condition. But to the degree that we believe that neurodegenerative conditions are being driven by dysfunction of the immune cells and supporting cells in the brain, and that dysfunction is in part caused by the fact that they have excess lipids in there. You see lipid droplets in Huntington's disease, for example, in microglia in the brain. So if you can do something about that, now, can you do something about that by just sucking cholesterol out of the liver and letting the body try and rebalance? That's unclear. We're not looking at the brain in our studies right now, but it's quite possible that will be a way forward to that. Or will you have to deliver it straight to the brain? You would inject into the cerebrospinal fluid, do a spinal tap and put the therapy in there, which is obviously much less pleasant than an IV introduction. And will require a different lipid nanoparticle and maybe a different delivery system. But this lies in the future, and the only way you can really say, is that gonna be really effective in humans, is to try it in humans, because the mouse models of Alzheimer's are really sort of kind of artificial. It's often very hard to say whether a result in a mouse is particularly relevant to what's going on in the human case, particularly where you're altering some aspect of metabolism. So down the line, I mean, after we've looked at liver disease, after we've looked at maybe some other rare diseases, yes, but the amount of suffering and the prevalence of these neurodegenerative conditions means that once we have proven efficacy in humans, people are going to try this. There will be research.
Patients who carry one copy of the ApoE epsilon 4 allele, which is involved in cholesterol transport, have a fourfold risk of developing Alzheimer's disease. And patients who have 2 copies have 12 times more risk of Alzheimer's disease. So there's something going on there in Alzheimer's, right?
Yeah, and like whether that the recent evidence suggests that microglia are strongly affected by the ApoE variants. And again, you can look at these diseases and see microglia that have lipid droplets. And maybe something can be done about this. Maybe it's prevention. Maybe it can actually help regress some of the existing pathology. But until you start to do the studies, who knows?
That's right. And you also mentioned liver disease, MASH, metabolic dysfunction associated steatohepatitis. What is the role of the cholesterol degradation platform here?
Well, here, this is the area in which researchers have long theorized that that the fibrosis in the liver is the irreversible part of MASH. It disrupts liver structure and function, and you can get thin, you can get religion, you can change your lifestyle, but the fibrosis is there to stay. You can't get rid of it once you've got it. which is the problem. That's the real problem with that condition. And
Mhm.
the most recently approved treatment by Madrigal Pharmaceuticals, resmetirom, you take that for a year at a cost of $50,000, and some patients see a 10% reduction in their fibrosis. And that's not a great result, but it's better than the nothing that existed before this drug was approved. In our mouse studies, we produced something like a 50% regression of fibrosis um, over 6 weeks of treatment. So like atherosclerosis, there is something about the free cholesterol presence in the liver that is inhibiting existing maintenance and repair mechanisms that would otherwise be fixing that disruptive fibrosis that is the characteristic of MASH liver tissue. So we believe that we can do some good there. It's just not a first priority.
Well, very, very interesting, but one step at a time.
Yeah, one step at a time, since investors tend to only fund you for one step at a time.
So, so let's shift gears into the longevity industry in general. Um, you are the founder of Fight Aging, which is arguably one of the most comprehensive newsletters on aging. What led you to start Fight Aging?
This was all started because I decided when I was first thinking, wow, aging is really bad,
Mm.
back in the day, and then I had to go around and do some research, and isn't somebody doing something about this? And yes, no, not really. But it took quite a long time to dig through everything to try to get a sense of what was going out there. So I thought, well, why don't I publish a website and like put all this stuff on there, and that will save somebody else from having to do all this work later on. And that kind of grew into a news site because I kept looking at things and things kept happening. And from there, it turned into Fight Aging, and sort of branched out into whatever I was doing at the time to help raise funding for various research efforts. And then suddenly 10 or 15 years later, there was an industry and I was investing in it. And I guess my superpower is that I can just keep writing about a topic for 20 something years, and show no signs of stopping at this point.
I read it every week. A good roundup. Recently, you wrote a very thought-provoking commentary on a paper by Dr. Olshansky's group titled "The Implausibility of Radical Life Extension in Humans in the 21st Century." So could you maybe elaborate on what is radical life extension and why you believe betting against radical life extension in this century seems unwise?
Well, let's say radical life extension is 20 years, something way beyond, what you can do with exercise. So the difference between normal healthy person and normal healthy person with optimal exercise is not 20 years. It's probably 5 to 10,
Mhm.
based on the epidemiology. So certainly there's a very wide range between obese sedentary person and optimally fit person, but a much smaller range between normal and optimally fit. So if we say that radical life extension is 20 years, then and if we think it's reasonable to believe that senolytics can add 5 years, then we need four different programs that are going to turn out to be as useful as clearing senescent cells. And that seems a reasonable thing to achieve over the course of 70 years. Um, I mean, the scientific view is if you don't have evidence for it, it's impossible, or at least you can't say anything about it. And the Olshansky paper, I mean, comes down to the end and says, yeah, we don't know, but clearly it would take a bunch of work if you want to see radical life extension this century. But if we keep going the way we're going, you're not going to see that, which has always been the case. I mean, if you project a trend forward from the era in which nobody was actually deliberately trying to do anything about aging, obviously that trend is going to be wrong at some point in the near future, because we have a whole industry that's like trying to produce therapies that deliberately target aspects of aging. And at some point, it's reasonable to expect that they're going to achieve this goal. And 70 years is a very, very long time. I mean, even given the fact that medical development is ridiculously slow because it's ridiculously regulated. It's still a long time. Just look at the difference between 1950 and now. The medicine of the 1950s, not so great for a lot of things.
And actually, to be fair, I guess the longevity industry really just started blooming like in the 2010s, around 10, 15 years ago.
Yeah, hasn't had a chance to approve any drugs yet, really.
That's right. So what is,
Nothing meaningful.
sorry?
Nothing meaningful anyway.
Mm,
Obviously, the people who go after the low-hanging fruit and
mm,
chase supplements and whatnot that aren't expected to do anything much, they got stuff out there, but that's irrelevant to the goal of producing genuinely new technologies.
absolutely. So what is the main bottleneck of the longevity industry today, in your opinion?
Uh, I mean, the usual point, it's funding.
Mm,
It's a small industry. So, but everything needs to be bootstrapped. Nobody's going to fund something to the degree that it should be funded before you've got like, proof of principle. So you have to bootstrap your way forward. And certainly we're at the billions of dollars of investment stage right now. So really at this point, maybe the bottleneck is actually not the funding and more the let's get the first few therapies through the incredibly slow regulatory process. So it's more just
mm.
a time of regulatory process chewing than we just need funding. Because it's going to be another 5 years, I think, before some of the first therapies are really out there in more than a very trivial way, and probably more like 10 years.
And talking about regulatory agencies, is there a role for clinical research on these kind of next generation therapies in alternative jurisdictions that have perhaps more lenient or flexible rules for testing in humans?
Well, it's kind of interesting that the whole question of what are we going to do with the regulation of medicine is occurring at the same time as, hey, look, we have a really, really impressive set of capabilities that maybe we can develop here. Maybe it's not a coincidence that they're happening at the same time, but they're not necessarily connected. So in a world in which you can spend $2 million to develop a gene therapy and deploy it in a clinical trial in, let's say, Roatan, and get data that shows that it does X, Y, and Z, you can't sustain a industry that takes, $50 million to get to the same place inside the regulated regions. That's too big a disparity. Something has to change here because the incentives to produce progress are tremendous. So there has to be some pressure put on regulators. Some pressure will come about as a result of this. I mean, companies can go produce meaningful data at a tenth of the cost in of the fully regulated environment. And at some point, a lot more will do that, and the stigma will go away slowly, and the regulators will have to adjust the way they approach the world to avoid being outcompeted. But this is all going to play out over fairly long time frames, I think. It's not going to be the case that suddenly medical tourism explodes and the world is different overnight. But I do think that medical tourism for enhancement therapies, such as those that slow aging or reverse aging, are going to be a whole different beast than medical tourism for sick people. Because there aren't that many sick people compared to people who are merely aging down into degeneration. You have like 100 times more people who are potential clients, and at that point you have a whole different phase of the medical tourism industry, and things will change. There will be much more professionalism, but this hasn't happened yet. The technologies have not percolated out to the point at which the inevitable consequences have become inevitable. So we'll see this play out over the next 20 to 30 years, I think. Something has to give. You just can't have a world in which it's possible to do something that costs $50 million for $5 million and something not change.
Great point, and I guess we will see. So besides this cholesterol degradation platform, what other kind of therapies in the longevity industry are you most excited about?
I think I'm most excited about senolytics, honestly. because this has the greatest potential for somebody to come along and deliver it to the masses, because there are working senolytic drugs that are clearly beneficial in the animal models, and there's plenty of human data for their use in other areas of other areas of medicine. So why isn't anybody out there doing a online prescription service for dasatinib and quercetin, and delivering it to very large numbers of people and obtaining data? Why aren't people running low-cost clinical trials to validate that this actually is really pretty good? And sometimes just because a thing is possible, it doesn't happen, or it takes too long to happen. But somebody needs to do these things, because half the people in the US who have atherosclerosis, maybe they don't need to have atherosclerosis as badly as they do. Maybe they should be taking senolytics.
And who should sponsor this kind of studies?
Yeah, I mean, philanthropists, essentially, because nobody else is going to do it. It's the finding a for-profit way to do it. Yeah, there is one, but clearly nobody's biting that quite yet. The groups that could do it are sort of holding off on senolytics for reasons best known to themselves. They're focusing instead on things like rapamycin. But I don't see that there's, other than size of effect, I don't see why they shouldn't be going after that.
Well, I guess we'll see. Only time will tell. So my final question is, what is the best advice you have ever received?
I suppose that would probably be to think about minimizing regret when you think about your future plans.
Yeah, that's very practical advice. What makes you regret the least?
Or what do you think is going to make you regret your least? You can always be wrong, but if you don't try, then you're more likely to be wrong.
Very good. So, thank you for taking the time to speak with me. It was really interesting, and all the best for repair and for fight aging.
Pleasure talking to you.
Hi again. If you found this video interesting and would like more people to see this kind of content, consider clicking the like button and subscribing to our channel. Thank you for listening.